Hello everyone, my name is Paula Reyes and I'm a PhD student at Universidad Nacional Autónoma de México, and today I'll be sharing with you our work entitled Overlapping Genetic Factors in Brain Morphometry and Parkinson's Disease Risk. So to start, let me tell you that Parkinson's disease is the second most common neurodegenerative disease. It has both motor and non-motor manifestations, including rigidity, resting tremor, sleeping disorders, cognitive impairment, among many others. And its molecular hallmark is the progressive loss of dopaminergic neurons in the substantia nigra. Besides that, PD has a very complex genetic background. And more recently, some studies have identified that there is also group differences in subcortical brain morphometry associated with Parkinson's disease, particularly in structures such as the putamen, thalamus, and the caudate nucleus. For that reason, the objective of our study was to explore the genetic association between Parkinson's disease and 10 brain structures by identifying specific shared regions of the genome involved in both traits, meaning Parkinson's disease risk and the volume of brain regions of interest, and to shed light on the biological mechanisms underlying the relationship between Parkinson's disease and brain morphometry, and to perform their biological annotation. To do this, we employ two datasets from genome-wide association studies. For Parkinson's disease, we employ a meta-analysis from individuals of European ancestry, from cohorts including the International Parkinson's Disease Genomics Consortium, 23andMe, and the UK Biobank. This yielded over 56,000 cases and 1.4 million controls for a sample size. And in the other hand, the GWAS from subcortical brain regions and intracranial volume was also meta-analysis from European ancestry individuals and included data for intracranial volume, brainstem, caudate nucleus, hippocampus, putamen, pallidum, thalamus, ventral diencephalon, nucleus accumbens, and amygdala. This had about 66,000 participants. We used a three-step pipeline. To do this, we estimated the genetic correlation between Parkinson's disease and brain regions of interest using a bivariate linkage disequilibrium score regression approach, and we employed the Bonferroni multiple testing correction to define statistical significance. Also, we performed a pairwise GWAS analysis to identify specific shared regions of the genome and to further delineate the genetic overlap between Parkinson's disease and the regions of interest in the brain. Finally, employing the FUMA platform, we performed functional annotation across the entire genome. Our results showed that there is a positive significant genetic correlation between Parkinson's disease and the volumes of brainstem, caudate nucleus, hippocampus, putamen, pallidum, ventral diencephalon, nucleus accumbens, amygdala, and thalamus at a whole genome level, but particularly the strongest genetic correlation between Parkinson's disease and intracranial volume. The pairwise GWAS results showed 210 shared genomic regions between Parkinson's disease and at least one of the 8 regions of interest. Here I show you a rough diagram of the approximate location of these regions, and in black boxes you can see the number of shared genomic segments, while the number of plausible genes, which are genes that are significantly associated at the gene-based association level with both traits, are displayed in white boxes. Furthermore, the results for function annotation showed 129 genes significantly enriched in Parkinson's disease and at least one region of interest. Here, I show you an ideogram that highlights shared genes between brain regions and Parkinson's disease. In this study, the regions that had a higher count for shared genes were the putamen with 28 genes and the caudate nucleus with 27 genes. Finally, we want to highlight the biological relevance of some of these shared genes between Parkinson's disease and the regions of interest. As you can see, many of these highlighted genes are shared between PD and the putamen and the caudate nucleus. These genes participate in important biological processes including neuroinflammation, autophagy lysosomal pathway, hypothalamic pituitary adrenal axis regulation, verbal numerical reasoning and cognitive resilience, vesicle trafficking, and mitophagy. Finally, we want to assess the limitations of this study. We have to be cautious about the generalizability of these findings, as they were yielded from data of European ancestry individuals only, and do not necessarily can be translated to populations of other ethnicities. Also, analyses performed here are dependent on the statistical power of the original genome-wide association studies. As a conclusion, we provided evidence of the genetic overlap between Parkinson's disease susceptibility and brain structure. In particular, we are providing evidence for shared genetic pathways involving chronic inflammation, hypothalamic pituitary adrenal pathway, mitophagy, and the disruption of the vesicle trafficking and autophagic pathways. Overall, we hope that this could shed light about the etiology of Parkinson's disease.